All right, today we're going to talk about the synapse, in particular the chemical properties of the neuron, right? So how do we first discover the synapse? I've sort of hinted at this, right? We knew, this comes from Sherrington, right? And we knew this idea that action potentials move very quickly, right? And what Sherrington realized is that at some point they have to cross between neurons, right? From cell to cell, and that he found an example where if it just had to go from one neuron to two, things seemed much faster than if it had to go from one to two to three. That the conduction of the action potential was much slower across all of that. And he hypothesized that at these places where the two cells connected, what we now call the synapse, that some slower event had to happen there. So that's that slower event to talk about today. Now there is a big history on the synapse, and it actually resulted in kind of a weird case for the 1906 Nobel Prize. So people knew, they had been looking at neurons, right, and they were starting to looking at brains and things, and they knew that there were obviously more than one cell involved there, but what the question was, what happens when one cell meets the next one? This was unclear. We didn't really quite have the tools, so they were sort of making guesses. And there's sort of two main camps here: the reticularists, which I've shown here on the left, which sort of, their head was sort of Camillo Golgi, who, if you've taken biology, you've probably heard of the Golgi apparatus. That's because he discovered it, right? He's a big name in the field. And he thought this idea was this big reticular network where all the cells they were individual cells, but they were really connected. They were kind of fused together where they met. And the other camp was led by Santiago Ramon y Cajal, we see over here. And his idea was really more that the neuronists, they called them, right? Because they believed in the neuron doctrine, that each neuron was sort of a thing by itself, and that there was a clear separation between them. And they so the key thing is that Golgi developed what is called the Golgi stain, which it's actually still used today by some people, right? Where you could really nicely stain individual neurons, and then that led to these nice drawings here. And Cajal, though, kind of took it the next step. He was the much better as sort of artist, if nothing else, and really could stain these neurons. And when he looked very closely at them, oh my goodness. And when Cajal took these really good techniques yet, and he looked very closely at them, he thought he could see these sort of distinct disconnects between the cells. So he ends up being right? This is the idea of the synapse, where they kind of run right up to each other, but there's a space, a synaptic cleft. Right? But in the end, Golgi developed the tools and had a hypothesis. Cajal had a good hypothesis. Nobody could really prove 100% who was right now. And sort of in a great way, and I think this really represents what I try and get across the class, the committee gave both of them the Nobel Prize. They both had a good idea. They knew that they were both onto the right track, and so they both awarded the Nobel Prize for their sort of work as scientists. I happen to think Santiago Ramon y Cajal is one of the greatest neuroscientists. I'm not alone on that. He's well celebrated, but I just like to show this picture to you. And this is sort of a picture of him in his younger days. He actually went to school to become a physician, kind of forced into it by his parents. Seemed pretty bored with it and took up science more as a hobby and became really good at it, right? And a large part, I think, is sort of artistic skill. If you can see here, right, these extremely fine drawings. A lot of these early neuroanatomical drawings, they're really traced back to Cajal and his amazing abilities to do so. Right. So I do want to mention one thing, though, at this point, is that Cajal is mostly right. Most cells have synapses, but much, much later, we discovered that actually there's an exception occasionally. Again, it takes place in the retina frequently. Is this idea of electrical synapses, whereby these two neurons, and they come together, and normally there's this space called the synaptic cleft, but in some cases they actually join together and make these little pores using gap junctions, right? So that things can actually freely flow between them, much the way Golgi hypothesized with his reticular networks. All right, so that's the historical context. The real base of today is what controls the start or stop of an action potential, right? the action potential flies down the axon, then it gets to the end of the axon, and now what? Right? So here's the basic summary, right? Some terminology here. The action potential is arriving from here is what it's denoted. So this is the presynaptic neuron, right, before the synapse, and this is the postsynaptic neuron. And so if we go through the order of events here, there's the action potential arrives, go to 2, we'll go into this next, and there's some other steps here, right? That's going to induce these vesicles here that are full of things called neurotransmitters, which we'll get to, and they are going to, they're just sitting here all pulled up, and if we look at this electron micrograph here, you can see these nice, right? These presynaptic clefts are filled with all of these little vesicles full of neurotransmitters just waiting to be released. Action potential arrives, boom, they come and they attach to the membrane down here, and then they fuse and they release the contents of neurotransmitters onto the next side, right? On the postsynaptic side, they bind to neurotransmitter receptors, right? A receptor for a neurotransmitter. And this then has the effect to either basically increase or decrease the voltage in the postsynaptic neuron, which can either lead to or lead away from a action potential. Here's another way to look at it, and includes another step that I think is important, right? Action potential comes down the axon, causes, what really happens here, and it's sort of shown here, is the idea that what it does is it causes release of calcium through these pores. These are voltage-gated calcium channels, right? So voltage goes up because of the action potential. They open up, calcium rushes in. That calcium is what tells these vesicles for a neurotransmitter to release their contents and then bind to the neurotransmitter receptors, right? There's a final step we'll talk about a little bit later where we've got a whole bunch of neurotransmitters here. We want to be able to turn these things on and off, and to do so, there's usually some sort of cleanup phase, basically, where this is sort of showing the idea that you can have the vesicles can take them back up and sort of suck them back up and be reused. There can be channels here that can sort of, or pumps that can sort of pump them back into the presynaptic cell, or there's cases where they can, an enzyme can come in and just sort of chew them up and degrade them so they don't work anymore. All right. As a somewhat quick aside, something I think is very cool, this burst of calcium, when you release there, is something that ends up being a very useful tool for neuroscientists. So I imagine I've mentioned how we can do imaging to see sort of what is active in the brain. And one of the key things is we've developed very good tools to measure calcium levels. Since this is a big burst, we can sort of easily see it. So if we use a particular sensor that now gets brighter in response to higher calcium levels, and we put it into those neurons, we can see which neurons are firing action potentials. Or which, right? So here's an example where somebody can look at an entire larval zebrafish brain. Not the biggest brain in the world, but hey, it's pretty good, right? And they can watch this zebrafish. So what they do is they sort of stick their head in a little bit of gelatin to keep it in place, and then use a microscope to image their brain. They're pretty clear, so this is relatively easy. And then they put it in sort of a VR circumstance where they're showing it images and giving it sort of puffs of air and try and make it guide through things. And what they're trying to understand is very simple things at this point, right? And it's like, how does a zebrafish decide it wants to turn left or right? You know? And so if we look on the right here, it's kind of a fantastic thing that they were looking at about 98% of all the neurons in a zebrafish brain simultaneously, right? And this is saying if it's turning right or left, and they can look at all these flashes, right? This is looking from top view, side view, and bottom view. And this can then tell you, what parts of the brain are active during these different behaviors, right? And, so what you're really looking at here, these flashes might just seem kind of colorful fireworks-like, but you're really looking at a zebrafish thinking, right? And if we can do that here, the hope is that eventually we can do with something more complex. We work from a mouse to probably a monkey to eventually humans. All right, neurotransmitters. We mentioned this briefly. This is what's released at the presynaptic cleft. There's a whole bunch of them. This is already a small table of the sort of major neurotransmitters. But let's focus in on a couple here. Again, no real need to memorize, but it's going to be useful to be able to talk about this, right? Glutamate, shown down here, is the most common excitatory neurotransmitter. This is a neurotransmitter that is, when it's released on the presynaptic side, it's more likely to try and cause a action potential in the postsynaptic area. GABA is the most common inhibitory molecule, trying to do the opposite. These other ones I'm sure you've heard of. Acetylcholine does a number of different things. It's what's at your neuromuscular junctions, the ones that this controls your muscles firing actually, but it's also important for restfulness and awakeness and attention. Dopamine, this is one that was very important for reward, for motor behaviors, but it's sort of one of those ones we consider one of the happy molecules, along with serotonin. Although serotonin has many other sort of things it does as well. It's important for muscle function and sensory as well. But we often think of it as the happy drug because this is where antidepressants work on, is serotonin. And then glycine has a wide range of uses. Is usually inhibitory, and norepinephrine is almost hormone-like, and is very much, it's basically adrenaline, right? That's one of its main functions. All right. In addition to neurotransmitters, I do want to briefly mention there are other molecules that are neurotransmitter-like called neuropeptides. These are bigger, and this table sort of nicely has, I think, a nice comparison I made between the two. The main thing is that as opposed to being real direct at one synapse, neuropeptides can sort of spread over a big area, affecting multiple synapses. They're much bigger, they're slower acting, and they tend to be, whereas a neurotransmitter is sort of like a yes-no answer, fire an action potential, don't. Neuropeptides can be a little bit more like, oh, you're probably more likely to fire an action potential if you kind of feel like it, right? They kind of gently make things go up and down, and because of this, they last much longer as well. All right. So, neurotransmitters bind. What happens in the next cell, right? Let's come back to these main ions of neuroscience: sodium, potassium, calcium, and chloride. Why don't you think through this? Which of these ions above do you think are associated with an excitatory synapse? A synapse, an action potential reaches the end, it releases something like glutamate, right? That's excitatory, and that's likely to cause an action potential. Which of these two do you think are most likely to be in that camp, right? To be involved in that process? What you're thinking about that, this process is called an EPSP, or an excitatory postsynaptic potential. This was in that graph for today. And on the other side, which ions do you think are responsible for an inhibitory synapse, or an IPSP, an inhibitory postsynaptic potential? All right. So hopefully you've had some time to think about it, wrote down your ideas. So the main thing is here, which ones of these are going to increase the voltage inside of that next neuron? So if you open a channel for sodium, that should be pretty easy. That's what causes an action. So that's excitatory. Potassium, that's a little tricky, right? It's positive, right? So you might have thought this one was excitatory, but there's more potassium inside the cell. So we know this, right? When you open the potassium channels, the voltage drops. So that's inhibitory. Calcium, you probably didn't have a good way to know other than if you thought about the presynapse, right? When we open voltage-gated calcium channels there, calcium rushed in, right? So that's what's going to happen here is this calcium with two positive charges are going to rush in, increase the voltage. It's excitatory. And last, there's chloride. So you maybe didn't have enough information, but could make a logical guess here based on its negative charge. It's going to end up being inhibitory. More chloride outside, you open up the chloride channel, it rushes in, makes things more negative. That's further away from the threshold that causes an action potential. These neurotransmitter receptors come in two flavors, ionotropic and metabotropic. We'll talk about this a little bit, but don't worry too much about the real big molecular biology of this. The main thing is ionotropic, they're the easy ones, right? Neurotransmitter comes in, binds to the receptor, opens it. Ions go, and it's a channel in itself. So this is an ion channel that's also a neurotransmitter receptor. So if this is a sodium channel, you just open the sodium. If it's a chloride, GABA binds and it opens, right? Metabotropic are basically receptors that can't do it by themselves, right? So there's a receptor here, this big pink thing, it binds to a neurotransmitter receptor. In itself, that doesn't cause anything that has ions, right? What happens though is that activates a protein, which activates another protein, which makes another messenger, which finally talks to the channel that now it'll say to open or close and let the ions in or out, is what it might be, right? So the main thing to think about is a little bit of, you should understand there's an intermediate process here, but the bigger thing is that this process here with ionotropic, fast, it's very fast, right? There's no intersteps. It's also shorter because of this and fewer ions than are able to get in and out, right? The metabotropic is much slower, there's more steps, that should make sense, but because it's longer, because of this, there's multiple things that have been turned on, there's a whole process of amplification, it has a much bigger response that's more longer lasting. Rod McKinrick, just a quick aside, is sort of the one who figured out, especially how ionotropic, but also how some of these other receptors have worked. He did X-ray crystallography to figure out their structure, both in replace of neurotransmitters and not. Sort of the first one in his lab to do that. The big point I mentioned him for is not necessarily bringing this up. I like it as a sort of picture of how you can do science, because here's the thing. He was a tenured professor at Harvard in the neurobiology department there, and he was doing something pretty different at the time. And he basically said, oh, I think I've got this idea that'd be really good to do this exact research. It was his proposal. And Harvard was like, oh, that's not what we pay you to do. You were doing good work. We gave you tenure. Why don't you just keep doing that? And he kind of got fed up with it and decided, I'm going to leave. And he goes to Rockefeller University and sets up his lab and does this. And he wins a Nobel Prize for it quite quickly, actually, right? So the point there being, breaking on your own can be a good thing. You don't have to, just because you would think you're tenured at Harvard, probably going to just stay set, right? I've got a good life. Can do mostly what I want to do. But sometimes, if you think you have a really good idea, you need to go for it. All right. So this is an example of some of his work. It's just really just showing, this is showing basically the shells and different ions, that are there of this. So if we're looking sort of top down at one of these channels, right? This is the acetylcholine ionotropic receptor. So this is the top, acetylcholine not bound, and then the bottom is showing it's bound. And it literally expands, right, to let those sodium ions in. And then there's a whole thing to talk about. How does the shape and the charges on the surface and everything decide that the sodium gets to go in or out, or what particular ion it's selected for. Here's a little bit more about the metabotropic receptors. Again, I'm not going to ask you to know too much more about that, other than the idea that there's amplification steps. So commonly they use something called G protein coupled receptors. So it binds to this G protein coupled receptor, which activates a G protein, which activates something like adenylate cyclase, right, which makes a whole bunch of things, which then in turn activates an ion channel, right? Just know that there's this multiple rounds that makes it slower, but there's also many more applications. At this whole step, one receptor can activate multiple G proteins, which can activate multiple adenylate cyclase. So you get this amplification, which both ramps things up and makes it slower to turn off. Here's a question for class next time, right? Why would we have two? Why is that functionally important? All right. All right, so I want us to think a little bit about how electrical properties might be different in different parts of the neuron, so that we can understand how you go from postsynaptic all the way through to starting the action potential to presynaptic side, right? So the axon is voltage-gated, action potential, all or nothing, whereas the dendrites are ligand-gated. Neurotransmitters are ligands, right? They're molecules that combine. There's a synaptic potential, and it's graded, right? You can have slight increases and decreases in voltage. You don't need to have this all or none response, right? And so if we go back to thinking about the axon hillock, what we talked about last time, right? In these dendrites out here, you're having all sorts of charges coming in. There might be a few negative charges, a few positive excitatory charges, and they're sort of, none of those are maybe huge in themselves, but they all add up to something, right? And so these ions are diffusing down, and eventually, right, they get to the axon hillock. And the whole thing is, you have to know there's that threshold, right? It's going to be a little bit different, but let's say it's minus 40 millivolts for a particular neuron. And do these charges change that voltage, those charges coming in from the dendrites, to get above that, or do they not? If they are above, bam, action potential flying down, right? Then it gets to the presynaptic side, releases neurotransmitters, whole thing starts over. All right. This is really just a summary, and I will leave this here for you for later. All right. So when you get to the dendrites, they have to do this thing, right, where they're going to decide eventually, should I fire an action potential or not, like I was just talking about, right? And this whole process, there's a few different ways, right? I always talk about the axon hillock does the math, sort of. I think I mentioned that in the slides. Right, so what it's doing is basically adding up charges. And here's sort of a depiction of how this might happen. Again, this is from your graph for today. Right, so the blue ones here are excitatory synapses coming in. The red ones are inhibitory. And you can see what's happening here. One of these fires, it gets you a little bit closer. Right here at negative 10 millivolts, that's the threshold. Closer, closer, almost there. Oh, then a red one fired, brings it down. And then this is showing that, nothing fired, and then so it's going to slowly make its way back to rest. Blue one excites, inhibits, and then a big excitation just gets above the threshold, bang, right? Action potential. And we call this temporal summation because this is looking across time, right? So this could just be a couple of these synapses. Maybe we're even just thinking about these two, and it's just looking over time, right? We could have, and this is trying to depict, there's also spatial summation, right? Where maybe you need, for a particular neuron, you need 3 simultaneous excitatory action potentials to come in, right? And so if they are each at a different space, right, that's the spatial part, different points in space, right, then they will eventually add up to cross that threshold and cause an action potential, right? And I think this sort of summarizes all of those together. You can get temporal summation is just looking, you can more simplify this. If you have one axon coming in, you're getting 3 quick action potentials, bam, 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 they all add up. Spatial summation, you might have 3 separate ones, but they're coming in about the same time, and they can also add up to roughly the same. Now, where these are coming in at does matter, and I think we'll talk about more of this next class, right? If they're closer to the cell body, they're going to add a bit more. You can have synapses that are stronger, right? They might be bigger or different things. So let's talk about that a little bit more. So this is an example of something called dendritic spines, where you can really, what we're seeing here are dendrites, right? And these wiggly things here, these are spines, and these are the postsynaptic sides. What you can't see is that on each one of these, there's another neuron that's come in and made a synapse on these dendritic spines, which are really the postsynaptic part. These are highly dynamic, and this is important because that can regulate the strength of the synapse, right? So if you can have one that's maybe got this really thin neck and is very long, that one might not be able to cause as much change because there's sort of a point here that's sort of restricting that, right? Or if it's, right on this bigger dendrite, it might cause a greater effect. Oops. All right. So we've till now shown fairly straightforward. Here's a couple synapses thing. I like this picture a lot because I think it is much more actually how this happened, looks like, right? Most neurons, they can have, thousands of inputs coming in, right? Thousands of synapses on the input side. And so this idea of making a decision or the axon hillock doing the math really gets more complicated. And this shows you also that, yes, you can have all these synapses on the dendrites, right? But you also have all these on the cell body. And the idea here is that, if you have a synapse right next to the axon hillock, that's a lot more impactful than a synapse way up here that any ions this lets in have to diffuse down this narrow thing, around the nucleus, and then eventually make their way here. Right? So I think let's just summarize sort of a little bit of what happens at the action potential, and we will call that it for today. Right. So again, I like this diagram. Kind of, I think, summarizes most of what we talked about, right? This is what happens at the synapse. Action potential comes in, right? That's going to increase the voltage at the presynaptic side. That's going to activate those voltage-gated calcium channels here, let a bunch of calcium in. That, in turn, will tell all of these little synaptic vesicles full of neurotransmitters to fuse the membrane and release their neurotransmitter into the synaptic cleft, the space between the two synapses. On the postsynaptic side, you have neurotransmitter receptors. Remember, they can be inotropic or metabotropic, and either directly or indirectly, they're going to cause ions to flow one way or the other. They're going to flow either into the cell or out of the cell, right? Out of this postsynaptic neuron. And then, depending on what ions are, those are going to cause a change in voltage on the postsynaptic side that will either help lead to or away from an action potential. And then the last step here is that at some point we have to clean up these neurotransmitters. A number of ways we can do that, but if we leave them alone too long, they're going to continue to activate on this other side. That actually, as a point, is sort of how basically how antidepressants work, right? Your SSRIs, right? Mostly what those are, they're reuptake inhibitors. So they're going to block this process and keep serotonin in the synaptic cleft longer, keep these synapses active longer, and that is known to help alleviate depression. So that is kind of where we're going. For next week, we are going to, we've really covered the sort of the base level knowledge, right? The very factual knowledge, and now we're going to try and apply that and understand it a little deeper by what happens when you perturb the system. And what I like to use a lot is to focus in on how do drugs and toxins work, because I think that's sort of an interesting way to understand this. All right. Thanks, and we'll see you next class.